Hello everyone, welcome to my channel Love of Biology. Today we are going to learn about Agarose Gel Electrophoresis. So this method is used to separate the biomolecules based on their size. So agarose, it is a solidifying agent which is obtained from the algae. So let us see. Electrophoresis is a technique used to separate the macromolecules in a fluid or gel based on their charge, binding affinity and size under an electric field. So this was first observed and developed by Ferdinand Frederick Rios. Electrophoresis has a wide application in separating and analyzing the biomolecules. So we know the biomolecules, proteins, plasmids, nucleic acids, nothing but the RNA and DNA. To separate these things, we will use which is very useful in RDNA technology. It is biotechnology. So let us see the process. Prepare the sample. DNA sample we have to prepare, which we use dye to absorb it. Prepare an agarose gel solution, which is prepared by adding the 1% of agarose gel and 100 ml of TAE to 1 gram of agarose. So we use this. So next, gel casting. Pour the agarose TAE solution in the casting tray to solidify it. And next, set up the electrophoresis chamber. Fill this chamber with this buffer. Place the solid gel in the chamber. So after that, load the wells with a DNA sample. Next, connect the positive and negative points to the power supply and chamber. Switch on the power and migration of the DNA sample due to the electric field will generate the negatively charged molecules. What are the negative charged? DNA. So they will move towards the positive charge ones. So once you see the migration of the color DNA samples, because we dyed it in the gel, switch off the power supply, remove the gel and place it in the ethidium bromide solution. So exposing this ethidium bromide stained gel under UV light and take a picture. DNA bands appear in the lane of respective well. Also the DNA ladder is visible. There the length of DNA bands can be determined. Below is the image if you see. So here this is the process. The wells in the first thing, later we have to pour the DNA samples which are with a blue dye here we used, and the migration, the bands also you can observe under this. So the bands also we can see based on the size, that is the larger molecules will move slower than the smaller ones. So smaller molecules will move faster compared to the larger ones. So based on that we can separate them. So what is the advantage? For most applications only a single component agarose is needed and no polymerization catalysts are required. Therefore agarose gels are simple and rapid to prepare. This gel is easily poured, does not denature the samples and recover. Thank you.